All right, so this is a pretty difficult topic that we're going to be going through. So if you do have any questions, please type them into the chat box and I will pause and answer them. All right, so first, just starting with the basics, what genetics is, it's the study of heredity. Genes are the chemical factors that determine traits. And then more specific, alleles are the different forms of a gene. So for an example, if we said the gene that we're looking at is the gene for plant height, then we would have alleles for tall or for short. And whenever we usually represent the allele with the letter, and the capital letter is the dominant form, and then we just use a lowercase letter for the recessive form. And we'll talk about what dominant and recessive means. So who discovered all of this and worked with to be the father of genetics his name is Gregor Mendel, and he was an Austrian monk. And in the monastery that he worked in, he was in charge of the garden. So he worked with pea plants, and he discovered all of these cool topics and a lot of the information about how traits are inherited. Okay, so pollination. So we said he's working with pea plants, and the pea plants had flowers on them. The flower is the reproductive part of the plant. So we have a couple different terms. Self-pollination is when the sperm cells, which are in pollen, fertilize the egg cells in the same flower. So we can see in the picture here that we have the male parts and the female parts. So the pollen falls into that same flower, and so it self-pollinates itself. And that means that every flower is going to be identical to the parent flower because there's no outside DNA. It's just that flower pollinating itself, so it's going to essentially be making like clones of itself. And then the plants that were allowed to self-pollinate, we call them true breeding. And we said they produce offspring identical to themselves. And then cross-pollination produces seeds that have two different plants as the parents. So Gregor Mendel actually, this can happen in nature. Pollen can be carried by wind or by animals, so we can get cross-pollination that way. But Gregor Mendel actually did a more controlled version of this. He removed the male parts of one flower and then removed the female parts of another flower. So then he took the pollen from one flower with like a paintbrush and he dusted it onto the female parts of another flower. So he cross-pollinated them. And so he knew exactly which plant he was taking the pollen from and which plant he was fertilizing. So he said when they self-pollinate, we call them true bred. When we cross-pollinate, we call them hybrid. So that's when they have two different parents as two different plants as their parents. So he looked at 7 different traits with the pea plants. So the traits are listed along the top here. And then all of these only had 2 different forms of that trait, a dominant form and a recessive form. So he looked at seed shape. The dominant seed shape was round, and the recessive seed trait was wrinkled. And so we said to use a capital letter and a lowercase letter, so we could do big R for round and little r for wrinkled. The color of the seeds, they could be yellow, which was dominant, capital Y, or green, which is recessive, lowercase y. The color of the pods, dominant was green, capital G. And Yellow is the recessive color. That would be lowercase g. The shape of the pods, they could be full, which is dominant. So I'm writing with the mouse, my handwriting's kind of bad. Or they could be flat or constricted. For plant height, they could be tall, or they could be short, and this is the one that we saw as the example on that other screen. For the position of the flowers, they could have the flowers at the leaf junctions, or they can have them at the tips of the branches, and at the leaf junctions is called axial, or where they're just at the tip of the branches is called terminal. Oops, that's an A. And then for the color of the flower, dominant is purple and recessive is white. Does anybody have questions so far? Okay, Tiffany, any questions? Okay. So Mendel's experiment was he crossed plants with each of the 7 contrasting characters, and he studied their offspring. So he took a purebred that was round, so one that was round and was self-pollinating, and then he took a purebred of the wrinkled. And whenever he crossed those, he found that the offspring were round, all of the offspring. When he did this with the seed color, he found all of the seeds were yellow. So we can see all the way down, he found that all of the offspring were the same, and they all possessed the characteristic physically of the dominant trait. So to his surprise, all of the offspring had the character of only one of the parents, and the other parent or the other characteristic kind of seemed to disappear. Okay, we're going to pause real quick. All right, so he did this first experiment, and it seemed very strange to him. He thought that the offspring would maybe come out as like a blend of both the parents, and he found that that wasn't the case, that all of the offspring had only the character of one of the parents. So the next step in his, okay, so the P here stands for parental. This is our first cross in crossing ones that aren't just self-pollinating. And then this F stands for filial, which is the offspring. So the first filial. So his next step was he took the first filial generation, and he allowed that to self-pollinate. Oh, sorry. So I jumped ahead a little bit. So the conclusions after he did that first test were he determined that biological inheritance is determined by factors that are passed from one generation to the next. And then the second thing that he concluded was that some alleles are dominant and others are recessive. So the dominant allele, if we have an organism that has a dominant allele, that's the one that we're going to observe. The recessive allele, an organism with a recessive allele will exhibit that form only when the dominant allele for the trait is not present. So a way that I think about this is if I were to come in wearing just like a light like fragrance, you would be able to smell it because it's on me. But then say the person next to me came in and sat down and they had like doused themselves in Axe body spray. That's the one that you'd be able to smell. It doesn't mean that my perfume disappeared. It didn't go away, but theirs like overpowered it. So that's the one that we could smell. So when the dominant is there, that's the one that's observed. So if we have a dominant and a recessive, the dominant is the one that's observed. So the only way that we can observe the recessive is if the dominant isn't present. The only way that you'll be able to smell the soft perfume is when that strong Axe body spray isn't there. So taking the next step, so what happened to that recessive trait? He found that it was still there, but it was being masked by that dominant allele. So the next step was he took the F1 generation and he let it self-pollinate, and he found that those recessive alleles reappeared. So because they reappeared, this indicated that at some point, the recessive allele separated from the dominant allele. And the term that we use for that is segregation. It's the separation of alleles during gamete formation. So here is our first filial. So we said that he did the pure breeding purple, in this case the P, purple, and a pure breeding white, and he crossed him, and all of the offspring were purple. But then whenever he allowed that to self-pollinate, we can see, so this is purple, there's the dominant and recessive. So we said when the dominant's present, that's when we observe, so it's purple. And when it's self-pollinated, we can see that the offspring are not all purple. Purple, 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 white. So when the F1 plant flowers are allowed to self-pollinate, the alleles segregate from each other. So each gamete carries only a single copy of each gene. And a gamete is our sex cells, so it could be a sperm cell or an egg cell. They carry only a single copy of the gene. So each F1 plant produces two types of gametes, a dominant allele and a recessive allele. So we can see that here, or here, a capital P and a little p. And then our F2 generation, we have a 3-to-1 ratio of purple flowers to white flowers. So Punnett squares are gene crosses or gene combinations that might result from a genetic cross, can be determined by drawing a Punnett square. And then Punnett squares, that's what these are here. They can be used to predict and compare genetic variations that will result from a cross. They can't determine the precise outcome, but they can determine the probability. So like here, if we were to cross a purebred tall with a purebred short, we get all four big T, little t, which means since tall is dominant, there are going to be tall. If we took two of those big T, little t and allowed them to cross, we can have a big T, a big T, little t, or little t, little t. So we can say that every time they have a baby, there is a 75% chance it's going to be tall. It doesn't mean that if they have 4 offspring, that 3 will be tall and 1 will be short. It means each offspring has a 75% chance of being tall. So he said it can't predict the precise outcome. It can't say if you have 4 children, they're going to be 3 tall and 1 short, but it can predict the probability. So we say if you have 4 children, each child has a 75% chance of being a tall plant and a short plant. And how we do our Punnett squares, we put one parent on the top and one parent on the side. And then this T and this T go in this box. So it's like if we were to move our fingers to, same as like when you put things in a data table. This T and this T go in the next box. This T and this T go in that box. And then this one and this one go in that box. And how we put them, arrange them, is if there's a capital T, we always put that one in front. So we'll never see little t, big T. It's just if there's a capital one, we put it in front. Questions? Hey, Cheyenne and Amber? Okay, so that's called a Punnett square. Some of the vocabulary terms, we have a couple different ones we use. Homozygous and heterozygous. Homo means the same, and hetero means mixed. So if both alleles are the same, it's called a homozygous. So homozygous can either be capital, capital, or it can be lowercase, lowercase, but they're both the same. So organisms that have two identical alleles for a particular trait. So if we were looking at tall and short, it could be big T, or little t, little t. So those are our purebreds. Heterozygous are organisms that have two different alleles for the same trait. So these are our hybrids. So this is when there's a big T and a little t. And then our next set of terms are genotype and phenotype. Genotype is the letter combination. So for our offspring, we could say they have a 25% chance of being big T, a 50% chance of being big T, little t, or a 25% chance of being little t. So that's the genotype, or in that case, with the percentages, it's the genotypic ratio. And then the phenotype is the physical characteristic, P for pheno, P for physical, what it looks like. So that's not giving the letter combination, it's saying what it would look like. So tall or short. So if we say there's a 1 in 4 chance of being short, that would be the phenotypic ratio. So genotype, letter combination, phenotype, physical characteristic. All right, our next trait is pedigrees. This also, we use our alleles, our letter combination. And this shows the occurrence and the appearance of phenotypes of a particular gene and its ancestors from one generation to the next. So in our pedigrees, a square means that it's a male, a circle means that it's a female, and then whatever trait it is that we're looking at, white means that it doesn't have that trait, and dark means it does. So these are A's. So on our list with Mendel's traits, that standard for if the flowers were axial or terminal. So you can see if the two parents, one of them was heterozygous, because there's a big and a little, and the other was homozygous terminal. So then we do Punnett squares to figure out the children that they could have. And I apologize, this is probably going to be messy. Oops. Okay, so we put one parent on the top of the Punnett square. And I put the other parent on the side of the Punnett square. And then we go ahead and fill it in. So this is big A, little a. What would this box be? Perfect, good. How about this one? Good. And the last one? Good. So with their children, we know they're going to be either little a, or big A, little a. We know this one's little a because it's colored in. We know these ones are big A because they're not colored in, and big A isn't an option. On the Punnett square, there is no big A. Make sense? Okay. And pedigrees are really useful when we're studying disease, because we can do these Punnett squares and we can trace it back with what we know and find out what the probability of offspring having that certain disorder is. Okay, so that's just, we call those Mendelian genetics when it's a dominant and recessive after Mendel, Mendelian. Whenever it isn't that way, it's not just so cut and dried, we call it non-Mendelian genetics. And we have a couple different ones we're going to look at. The first is codominance. So this is when neither of them are dominant, they're equally dominant. So we see this in cows, that if we cross a red cow and a white cow, we don't get all red cows or all white cows. We get this codominant. They're both expressed, and so we get this red and white cow, and it's called roan. So since neither one of them is dominant over the other, we don't use capital and lowercase letters. We use capital of both of them. So this question says, what offspring would be produced if a red cow, so if it's red, we know it has to be RR, is crossed with a roan cow. Roan cow is RW. So we put one on the top and one on the side. And we cross them. So big R, big R, big R, big R, big R, big W, big R, big W. And because these are different letters, we said when they're Mendelian genetics, we put the capital first. But when we have these ones, we just put them alphabetically. So that's the reason we put the R in front of the W. So the offspring for this situation here can be red or they can be white. Nope, not correct. They can be red or they can be roan. Sorry, that's a typo. But we're not going to have any white cows. That's not possible. So if we saw a white cow, we would know it didn't come from these parents. So codominance, they're both equally dominant, so both alleles are expressed. Not red, not white, both. Then our next type of non-Mendelian genetics is called incomplete dominance. And this is where one is dominant, but as it says, not completely. So in this case, we get a blending of the two traits. So if we were to cross a red and a white, we'd get a pink. And one instance where we see this type of heredity is with snapdragons. So once again, because it isn't just one is dominant over the other, we don't use the capital and the lowercase. In this case, we use this little apostrophe, and it's called a prime. So RR is red, R prime is white, and R prime is pink. So we see a blending. So it says, what would you expect if you crossed a white snapdragon, which is R prime, with a pink snapdragon, RR prime? And we look at our results, RR prime, pink, RR prime, pink, R prime, red, R prime, red. So we would say that our offspring have a 50% chance of being, I'm sorry, that's not red, that's white. I'm so sorry, guys. Okay, so they have 50% chance of being pink and they have 50% chance of being white. None of them are going to be red. So this is incomplete dominance. So the red is dominant and the white is like they blend together. Okay, the next type of non-Mendelian genetics is called multiple alleles. With what Mendel did, there was only 2 alleles. For plant height, there was tall or short. For flower color, there was purple or white. There weren't any others. That isn't the case most of the time. The majority of our traits are a lot more complicated than that. One instance is with our blood type. The gene has, for it to be considered multiple alleles, the gene has 3 or more possible alleles. For blood type, we have IA, which is type A. So if it's IA, it would be type A. IB would be type B. Those are dominant to type O, which is little I. And then A and B are codominant. So if it was IB, it would be type AB. Let me list these all out, sorry. Here, let me type them. And I can't make superscripts, so it's just going to be, I'm just going to type A. So AA is type A. BB is type B. OO, which is the little I, is type O. AO would be type A, because the O is recessive. BO would be type B. And then AB would be type AB. So A and B are codominant, and then A is dominant to O, and B is dominant to O. So it says, what are the possible genotypes and phenotypes for a cross between a type A mother and a type O father? We know type O because O is recessive. It has to be little I, little I. So, oops, little I, little I. But for the mom, because it just tells us she's type A, it doesn't tell us if she is homozygous type A or heterozygous type A. So we have to consider that she could be either. So we'd have to do two Punnett squares, one where she's homozygous and one where she's heterozygous. So if we cross those, we can see in this case, the offspring, would this be type A or type O? It would be A because A is dominant to O. So this would be type A. This would be type A. This would be type A. And this would be type A. And then over on the other Punnett square, this is type A. This is type O. This is type A. And this is type O. So if it just told us that the mother was A and the father was O, we could say that the child could be type A or type O because we don't know if the mom is homozygous or heterozygous. If the mother had, 5 children and they were all type A, we could probably kind of assume that she was probably homozygous. But still, you wouldn't be able to be 100% sure unless you blood typed her. I know this is a lot of information. Do we have questions so far? Oh, that's good to hear. Okay, so next, this is still Unit 5, but this is the section on DNA and RNA, and this is the one that we're going to have some practice on. I've embedded a lot of additional practice worksheets, or not embedded, linked into the course, and this is going to be what your alternate discussion is on in Lesson 24, so on Thursday. Okay, so first, just looking at the structure of DNA. We've looked at this before. It's a double helix, and each of the individual units that make it up is called a nucleotide. So here's one nucleotide. A nucleotide consists of a 5-carbon sugar. In DNA, the 5-carbon sugar is deoxyribose. That's the D in DNA. And in RNA, the sugar is ribose. That's the R in RNA. The nucleotide always has a phosphate. And then it also has a nitrogen base. And in DNA, those nitrogen bases can be adenine, thymine, cytosine, and guanine. And so we usually just use the capital of the first letter to abbreviate them. So A, adenine, T, thymine, G, guanine, C, cytosine. And we can see in DNA, it shows us nice and neat with how they're shaped here, that they can only fit with one other one. The C can only fit with the other G. So think of them like puzzle pieces. A C couldn't fit with another C. A C couldn't fit with an A or a T. So in DNA, A always pairs with T, and G always pairs with C. And this is going to be the same for RNA, with one exception. We're not going to have T's, we're going to have U's instead. Let's see how they pair together. So more looking on a nucleotide again. So the individual unit of a nucleic acid is a nucleotide. We said there's a 5-carbon sugar. For RNA, it's ribose. And for DNA, it's deoxyribose. And we can see that they look very similar to each other. And then the phosphate group is just a phosphate group. That's always the same. And then we have the nitrogen bases. And we can split our nitrogen bases into pyrimidines and purines. It's just how they fit together. We saw in the other picture how it was like puzzle pieces. A purine and a pyrimidine fit together to make a connection. So it's always cytosine and guanine. Those two always pair together, and we can remember that because the C kind of looks like a G. So that doesn't matter if it's DNA or RNA. And then we always have adenine. And then we see a difference, though, of what adenine fits with, depending on if it's DNA or RNA. In DNA, A fits with T. In RNA, A fits with U. All right, so we have two different processes. We have three different processes. DNA replication, transcription, and translation. In DNA replication, it's exactly what it sounds like. We're starting with one piece of DNA, and we're replicating it so that we end up with 2 pieces of identical DNA. And how this happens is the DNA unzips, just like a zipper, so that we can use one half of the original to serve as a template for the new half. So it unzips, and then we have these little exposed DNA bases along the zipper. And so that's how the new nucleotides coming in know where to line up. If there's a thymine on this part of the DNA, on the original part, then an adenine nucleotide will pair up there. If there's a guanine nucleotide on the original piece, a cytosine nucleotide will come and pair up. And so the nucleotides, they come in and they join one at a time based on what's complementary, A to T and C to G. And then we have this little guy come along that seals them together. And so what we end up with is 2 pieces of DNA. One with each has half of the original strand and half of a new strand. All right, I have a video on this that I'm going to have you watch. It is a little bit long, but I feel like it's worth watching. So it's almost 8 minutes. So I'm going to go ahead and put that in the chat box. So watch that and then click done once you're finished. Okay, so do we feel pretty comfortable with that topic? All right, so the next step, so that was replication. Our next process is called transcription. It's transcribing. So if you were to transcribe, you're kind of taking a like an account of it so that you can share it with somebody else. So it's this RNA is made because the DNA is trapped in the nucleus. It's like a prisoner there. So since the DNA can't leave the nucleus, the only way that it can get its message out of the nucleus is to get it to a messenger. And so the process is kind of similar to the replication in the sense that the DNA unzips and then the nucleotides fill in, and that's how they know where to fill in is by base pairing. And what we're, in this case, how it's different is that we're only making a single strand mRNA. And it's mRNA, so we do have a couple differences. The sugar is different. It's ribose instead of deoxyribose in the nucleotides. And then in place of thymine, we have uracil. But these nucleotides bind with their base pair, so we're making this growing chain of mRNA. And then once it makes the mRNA, it's told it the message, then the DNA zips back up, and the mRNA can leave the nucleus, and it can travel to the ribosome. So this is how the DNA, which is a prisoner in the nucleus, gets its message out of the nucleus. So we said they're base pairing, but because there isn't, if we have an adenine, sorry, hold on. If we have an adenine, because there's no thymine, it's going to pair with a uracil. If we have a thymine in the DNA, it still pairs with an adenine, and then there's no change in the guanine and cytosine. So this right here is where we're going to see the difference, is if we have a if we have an adenine, it's going to pair with uracil. So then the DNA zips back up, that mRNA, and we call it mRNA because it stands for messenger, because that's what it's doing. It's taking the messenger from the DNA in the nucleus, and it's taking that message out of the nucleus. So that process is transcription, going from DNA to mRNA, messenger RNA. And then the last process is called translation. When we translate something, we're changing languages. In this case, we're going from nucleic acid language, DNA and RNA, and we're going to protein language, which its letters are called amino acids. Where our nucleic acid letters were called nucleotides. So this is our mRNA. So this is that message that it came from the nucleus. And it's going to go to the ribosome. And in the ribosome, these little guys that are called tRNAs, which stands for transfer RNA, or I think of them as taxi RNA because they kind of are like little taxi cabs. They are each bringing an amino acid, which is the little ball on the top. And they're pulling up to the mRNA, and they know where to park because of base pairing. C pairs with G, U pairs with A. So once they find where they match, so this one's U, 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 so it has to, U, 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 so it has to pair with A, A, A. So that's where it knows where to park its taxi cab and let off its passenger, which is its amino acid. And then once it drops off its amino acid, then it leaves, and it can go and pick up another amino acid. And we're making this growing chain of amino acids, which once the chain is done, it makes a protein. A chain of amino acids is called a protein. Okay, and I have a video that you can watch if you want to after we do some practice. It's like 15 minutes, so I don't want to take 15 minutes of your time right now. But we're going to do some practice. Do we have any questions before we do practice? Okay, so I'm going to give you a link to a Google document. So go ahead and log into that. Okay, so there's one of you, 2, 3. Good. Let me share my screen. All right, so you can type on the document. Don't just go crazy and start typing everywhere, though. So what it gives you is the DNA. We're going to do a couple practice together. So it wants you to write the mRNA. We use the DNA to get the mRNA. Then the tRNA. We use the mRNA to get the tRNA. And then the amino acid. And we use the mRNA sequence to get the amino acid. And we use this table down here. So if our if our DNA was TAC, we use base pairing. So T pairs with A pairs with either T or U. Would it be T or U in mRNA? So we said RNA, good, it would be U because RNA doesn't have thymines, and then the C would pair with the G. So that's our mRNA sequence. Then we use our mRNA to get our tRNA, so we do base pairing, so A pairs with U pairs with A, G pairs with C, and then we use our mRNA. This is the biggest place we get mistakes is people use the tRNA instead of the mRNA. We use the mRNA, AUG, and we use the circle chart. And how the circle chart works is we do the first letter is the innermost circle, so we go to that green A. Then the next letter is our next circle, and then the last letter is that outermost circle. So off of the A, we go to a U, so we go to the red off of the A, and then off of the U, we go to the G, which is that little yellow one, and it says it's methionine, M-E-T, our start. So we put M-E-T. So we're going to do a few more together, and then I'm going to give you a few to do on your own, and that's how you will earn your points. So if we have CGC to mRNA, we do base pairing. So C pairs with G, G pairs with C, C pairs with G. Then next we do our tRNA. Once again, base pairing off the mRNA. So G pairs with C, C pairs with G, G pairs with C. And then next we do the amino acid. We use the mRNA, which is GCG. So we start in the innermost circle, G. Off of that, go to the next circle, C. And off of that, go to the next outermost circle, G. And what amino acid do we give? Okay, so GCG. So we do G in the innermost circle, and then off of there, C. So it's like the periwinkle one. And then to the next G, so it would be ALA, alanine. Whoops. Okay, so the circle chart, you start in the innermost, and that's your first letter. And then off of that first letter, you go to your next letter. And off of that letter, you go to the last one. Okay, so let's do two more together, and then I'll give you a few to try on your own. So T pairs with A, C pairs with G, C pairs with G. Then our tRNA, we use our mRNA. So A pairs with, since it's RNA, it goes with a U. My fingers were on the wrong letters. Then G pairs with C, and then another G pairs with C. Then we take our mRNA and use the circle chart. So if mRNA is AGG, start in the middle with A, and then off of the A, go to a G, and then off of that G, go to another G, and that amino acid is ARG. Okay, so let's have, so I only have two of you logged in. So who are the two logged in? So if you can type it in the chat box in the Adobe room, if you're in there. Okay, so you must have lost Cheyenne. Okay, so Tiffany, oh, you're in there too? Okay, it only shows that I have two of you in the, oh, your Google Drive isn't working. Okay, can you see what we're doing? Okay, so if I assign you ones, can you type your answers just in the chat box? Okay, so Manice, I want you to do the next two that are highlighted. Then Cheyenne, you're going to do the two after that. And then Tiffany, you're going to do the last two. And it's okay if you make mistakes. We're going to go over them. So for now, just do the best that you can. So Tiffany, you can type yours, just go down. So do like this one, then that one, and then that one, and then I'll type them into the document for you. Okay. So this first part is base pairing. So we have our four bases, A, T, G, and C. We know C always pairs with G. It doesn't matter what, if we're going from DNA to DNA or DNA to RNA. So if we have a C on the one side, it'll pair with a G on the other side, on the other strand. And then, so that means you can, since all we have is G's and C's in this one, you can go ahead and do that one. So if it's GCC, what would that pair with? The G pairs with a, good. Okay, so then we can go ahead and do the next box. You're going to do the same base pairing, just backwards. So C pairs with, you're right, keep going. Good. And then next, we're going to use the mRNA, which is the teal one, or aqua, or whatever color that is, and we're going to use the circle chart. So in the first circle, we're going to do our C, and then off of that C, we go to a G, and then from that G, we go to the next G. Perfect. Good job, Manise. So then the next one, we still have C's and G's, but whenever we have RNA, we don't have T's. So a T will pair with an A, and then an A will pair with a U. So if we have G, T, C, so no, you use a U instead of a T. There still is A's. So in this case, in this first row, we don't need to worry about it, but in the next row we will. Because there's not a T in the mRNA, we won't have to type anything different. It would just be base pairing. But when we get to the tRNA, okay, so there's still pairs with an A, so instead of a U, that would be an A. Just when we go to the next one, oops. Yep, so CAG, good. Okay, then the next one, where it would seem like we'd go back to T, there's not T, so it'd be a U. Okay, that one's backwards. So if we look at here to here, that would show that a C pairs with a C, and we know that can't be right. Okay, so good job, Denise. So here we have A, and so an A pairs with either a T or a U. In this case, since this is RNA, it would be a U. So A pairs with U. Whoa, what just happened? Oh, well. Okay, so A pairs with U, and then a C always pairs with a G. So C pairs with G, and we can remember because they kind of look like each other. And then a T always pairs with an A. So that would be UGA. So Tiffany, tell me if I'm helping at all. Then the next one, we use this one to go to our next box. So U pairs with an A G pairs with a C, yeah, I didn't, Cheyenne, I tried to undo it, but it wasn't me that did it. So whoever did it would have to undo it. And then an A would pair with a U, Tiffany. So is that making sense, kind of, Tiffany? Okay, so we just have to remember the letters that go together. So an A pairs with a T or a U. It's a T if it's DNA, it's a U if it's RNA. And then no matter what circumstance, a G always pairs with a C. C with G, G with C. Okay? So here we went A, we said to either T or U. Since this is mRNA, it went to a U. Then C always goes to G. And then T always pairs to A. And then we used this one to go to the next one. So U to A, G to C, A to U. And then the last one's the one that's a little trickier. We use the blue box, and we're going to use the circle chart. So if we have UGA in the circle chart, we start with the U in the middle. And then off of that U, we go to the G. And then off of that G, we go to the A. So in this case here with UGA, it would go to the stop codon. And whenever we hit the stop codon, that means this protein is done. And so stop just means like little scissors, cut the chain of amino acids, and it's finished. Okay, so let's take a look at what we did. G-arginine. That one's correct. That one is correct. Okay, so this one is incorrect. We want to try that one again. And this is where it gets tricky because if you don't go off the mRNA, you get the wrong amino acid. And then this one's incorrect. So the ones that I'm deleting, if we want to fix those ones. Okay, so let's fix those ones and then we'll double check it. And then if we're feeling okay, then I have a couple more that you're going to do on your own. Okay, good. 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 Okay, so Tiffany. Yeah, that's seriously happens a lot of the time. That's the most common mistake. All right, so Tiffany, if you want to go ahead and do ACC. So if we have ACC, that would pair with no, so A, this is DNA. So if an A pairs with either a T or a U, depending if it's RNA or DNA. And then we're looking, this second row is RNA. So A would pair with a U. Then the C would pair with a G. and then another C would pair with another G. So we're going like down the row. Okay, so then we have UGG, so what would that pair with? U pairs with good. And then we use the mRNA and the circle chart, and what would UGG? What amino acid would that be? Good. All right, so I think we got it, so I'm going to assign you a couple more for practice. We're going to go down to the next page, and let's have Cheyenne do the first two, Tiffany do the middle two, and Manice do the last two. Okay, so perfect. You guys all got them correct. So do you feel comfortable doing this? Okay, good. This is the hardest part of it. Good. So this is exactly what you're going to be doing on, okay, and there are a lot of worksheets that there's a worksheet that you can practice, and then there's an answer key worksheet so you can see if you got it right or not. And I think I put like 4 or 5 of those in the course. And then this is also exact thing is what you're going to be doing for the discussion for lesson 24. Okay? All right. So that is the end of our synchronous session today. If you have questions, stick around and you can ask them. If not, you are free to go since you attended today and you participated, Manice, Cheyenne, and Tiffany. The 3 of you will not have to do an exit ticket. Amber, since you didn't participate, you will still need to do an exit ticket. And those of you that are watching the recording will need to do an exit ticket. All right? Thank you. Have a good day. Bye-bye. Have a good night.